So before we start talking about molecular biology of the cell and cell function and structure and so on, what I want to do is introduce you to what are called experimental model systems. These are basically organisms, model organisms that are used for molecular studies and to try and understand how cells function in an organism and how they contribute to an organism. Of course, everything that we learn about, everything that I lecture about, what you read in the book they're all done by experiments that people do. And quite often they will use these model systems in order to answer specific questions. And as you'll see there's lots of different model systems that are used, and some of them are kind of strange organisms, but they're used because they can answer very specific questions. So let's take a look at that. So again, model organisms, what are the purpose of using these things? And it's to define genes and components that are involved in cell processes. Cell division, cell differentiation into other cell types, cell adhesion, developing into tissues, cell development, cell behavior, all kinds of diseases. You can't really do a lot of these things using single cells growing in culture, which I'll talk about as well. So people use entire organisms in order to look at it in a more complex way. So I want to walk you through a series of organisms that are used quite commonly. Okay. So the first one that I want to talk about is one that probably everybody has heard of. It's very common. It's E. coli. It's a bacteria. It's a prokaryotic organism. No nucleus. The ribosomes are a little bit different than ours. The structure of the DNA is different. But these are bacteria, and one of the reasons that there's a number of reasons why they're used as a model system, and that is because, first of all, they're very easy to grow, extremely easy to grow. They're very cheap. The media that you need to grow them in is very inexpensive. They grow very rapidly. They can divide every 10 to 15 minutes. So they're very easy to grow. And in fact, this is probably one of the first model organisms that was used very early on in biology and molecular biology, mainly because they were so easy to grow. The entire genome is sequenced. It's 4,400 genes. And the truth is that many of the most basic and important processes that cells carry out were first described at a molecular level using E. coli. So the genetic code, for example, was established using E. coli. DNA replication, the mechanisms using E. coli. Translation into proteins, protein synthesis. All these basic core phenomena were described and analyzed using bacteria. Now we know a lot about them. We've been using them for years. We know a lot about their genetics. We can introduce genes very easily into E. coli. They're called transgenic E. coli. And in fact, many drugs that are used are produced in E. coli. Proteins like insulin or growth hormone, rather than purifying them from some source, let's say from cow liver or tissues, they're grown in E. coli. So the human gene for insulin has been introduced into E. coli. The human gene for human growth hormone has been introduced into E. coli. And you can grow large amounts of these things, and they will produce and secrete the insulin or the growth hormone into the media, and then it's purified from there. And that's what people use. So if you are a diabetic, or you know somebody who's diabetic and they inject insulin, that's a protein, and if you look at the bottle of the insulin, most likely what you'll see is it'll say R insulin, small r insulin, and the small r stands for recombinant. So these are recombinant, genetically engineered bacteria that are producing these compounds, these proteins for us. Now there are organisms that are even smaller than bacteria. and that one is shown here. This is a virus that infects bacteria. This is called a bacteriophage. And the bacteriophage have also been used quite extensively in genetic work for quite a number of years, although they're not used as much now. 10 to 20 genes, so a small number of genes. It's an interesting structure. So at the top there's a head-like structure, and the DNA, the genes, the DNA is contained in that head. Then there's a long column that comes down from that head. And then there are what look like legs, and they actually are kind of like legs. It almost looks like some kind of a lunar landing craft that attaches to the bacteria. And the way these things work is they're in the water. They could be in the pond water or sewer water. They're all over the place. And what they'll do is they'll attach to the bacterial, the surface of the bacteria using those legs. And then they will kind of crouch down and insert that column through the bacterial cell wall, through the cell membrane, and inject the DNA into the bacteria. Once the viral DNA is in the bacteria, it begins to replicate, and then it uses the machinery of the bacteria to replicate its DNA to produce more proteins. It's very efficient, and in a very short time, maybe 20 minutes, the cell dies and hundreds of new virus particles are released. It's actually shown here. Here's an infection of E. coli by a certain bacteriophage called T4. And what you can see is the bacteriophage are attached to the surface of the bacteria. There's a lot of them attached there in green. And then the bacteria itself is in blue. And what happens is these virus particles attach by those legs, and then they insert that column into the bacteria, and they inject the DNA. And as I said, they replicate and produce new virus. In this case, you see a lot of bacteriophage attaching to the surface, but only one of those, only one of those will be able to insert its DNA in. Once one is inserted in, somehow the others are prevented from inserting the DNA in. So one inserts, the replication ensues, and the virus particles are generated, and then you can see they're released at the bottom. The cell basically explodes. Now, the bacteriophage have been used actually as a model for regulated gene expression. So as I said, the bacteriophage has 10 to 20 genes. Once it infects the bacteria, and it takes, let's say, 20 minutes or 30 minutes for the new viruses to be completed, the genes are expressed in a very sequential manner. And so people who are early on interested in gene expression and how gene expression is regulated took advantage of that sequential series and looked at some of the mechanisms of that, and a lot was learned using those bacteriophage. Now we move into, rather than looking at bacterial or prokaryotic cells, we're looking at yeast, which are eukaryotic cells, in a way very similar to our cells, mammalian cells. They have a nuclei, they have ribosomes which are identical to ours, and all the processes are very similar. The regulation of cell division is really extremely analogous to how human cells divide. And again, the reason why the yeast are so popular as a model system is because they are easy to grow. The media is very inexpensive. You can grow them in a flask, shake them. They grow very quickly. So they're very easy to grow, very easy to manipulate. It turns out that in terms of, let's say, cell division, the mechanisms that control cell division in yeast are virtually identical to the mechanisms that control human cell division. So you can use yeast and ask questions about how cell division is controlled and learn about how human cells are controlled. And in fact, it's so similar that human genes can replace yeast genes and the yeast will work just fine. So in other words, you can make a mutation in a particular gene that would be required for cell division, so the cells can no longer divide, and then introduce a analogous human gene into the cells, and the pathway picks up just fine. The proteins work just fine. They interact with what they need to interact within the cell. And so we can learn about the function of human genes by looking at how they function in yeast. So a very important model system. And there are, in case the whole genome has been sequenced, and there's 5,770 genes, all of which have been identified. Now there are things that are like there were the bacteriophage that infect bacteria. There are also viruses, of course, that infect human cells or mammalian cells or eukaryotic cells. The one I'm going to tell you about is SV40. SV40 is a virus that's been studied for well over 50 years. We've learned a lot using this virus. This is a virus that infects monkey cells. So it's SV40 stands for simian virus 40. It infects monkeys. Monkeys get some kind of a respiratory illness, which they recover from pretty easily, like a cold. And you can take the monkey cells and grow them in culture, and infect cells with SV40, and the cells undergo very efficient replication of the virus. So people have used SV40 as a model system for DNA replication. Since the viral DNA replicates in a mammalian cell, and there's so many virus particles replicating at one time, then it becomes a little bit easier to study the replication process because so many are happening at the same time. So people have studied this and learned a lot about eukaryotic DNA replication versus the prokaryotic DNA replication and which proteins are involved and so on. Now, it turns out that this SV40 virus is even a little bit more complicated and has been useful for other things as well. And that is, if you take SV40, like I said, if you take SV40 and you infect monkey cells, it'll replicate the virus and kill the cells. If you take SV40 and you infect human cells or you infect mouse cells, it doesn't kill the cells. It causes them to become cancer cells. It causes them to become tumorigenic. So rather than killing the cells, it creates tumors. In fact, you can take SV40 virus and infect mice or inject it into mice, and the mice will develop tumors. So this was discovered in the early 1960s, and so it became a model system, really one of the first molecular model systems for how cancer develops. Even though it was not a naturally occurring virus in humans or in mice, the fact that it caused cancer allowed it to be used as a model system. So it was considered a tumor virus, one of the first known tumor viruses. And in fact, the first oncogene, actually, a gene that causes cancer, was identified in SV40. It was called SV40 large T antigen. And we've learned a lot about cancer through SV40 large T antigen because even though it's not an analogous system, it still triggered that process. And we've learned a lot about what proteins are involved in cancer through the SV40 large T antigen. Now, above and beyond that, there's an interesting historical event that took place. And that is, in the 1960s, early 1960s, there was a polio outbreak. People are probably aware of that. And a vaccine was developed against polio. The way those vaccines were developed was the polio virus was grown in monkey cells. And it replicated well in monkey cells, and the virus that was produced was inactivated, and that was used as the vaccine. In those days, rather than injecting it, I think early on people would drink it. It would be in a little tiny cup with some sugar. You drink it. That's the polio vaccine that I took. It was a little, like on a cube of sugar, and you drank it. Well, at about that time when it was being used, it was the time that it was discovered that SV40 caused cancer in mice, and that it was a potential human tumor virus. And it became clear at that time, or it was discovered at that time, that the monkey cells they were using to produce the polio virus were contaminated with SV40. They were producing SV40. So people who were taking those early vaccines, me included, were also being infected with SV40. Now, as soon as they made that connection that it could be a potential human tumor virus, they immediately stopped using the monkey cells as a way to produce poliovirus, and they moved to chicken eggs, which is what are still being used. So these viruses the vaccine is using inactivated virus. Same is true with flu virus. The flu virus is grown in chicken eggs, and then the virus is inactivated, and then that's what's injected. It's a little bit different than what's happening with the coronavirus vaccine that people are producing now. They're not doing it that way. They're not using the inactivated virus as a vaccine. I'm not sure why. But what they're using instead is they're taking RNA from the virus and injecting that into the organism, mouse or monkey or human, and then that RNA is expected to be producing the protein of the virus that then your immune system would react against. So it's a completely different strategy. But in any case, it turned out that the SV40 virus was contaminating the poliovirus vaccine, and then they found out that it may cause human tumors because it caused the cells to become tumorigenic, and they stopped using them. And there was always, for all these years, people have been wondering, could it have given rise to cancers in some people? Could the SV40 that contaminated this vaccine end up being responsible for some human cancers? Well, it turns out in the last, I'd say 10 years or so, people have been doing genetic studies of different cancers and in certain brain tumors, and I think also some lung cancers and a few other types of cancers. What's been found is in doing the genetic analysis of the tumors, they find SV40 DNA. SV40 DNA in these naturally occurring human tumors. Now, there's no real good way, and of people of that particular age, there's no clear way how people can be infected by SV40. It's a monkey virus, not commonly encountered. So chances are they did get the virus through these vaccines, ended up in these cells. Now, whether or not it caused the cancer is a completely different story. We don't know that. All we know is that the SV40 DNA is in those tumors. But that's about where it stands. But it's clear that there was a contamination, and it may have been responsible for some cancers down the line. Okay, so let's look at another organism that's used. This is a kind of a strange but very interesting organism. It's an organism called Dictyostelium. Dictyostelium is a soil amoeba. It grows in the soil. And it's been used as a model system for the following reason. You have, and you can see on the left-hand side of the slide, so you have these single-cell amoeba growing in the soil. And under conditions of starvation, that is, when the nutrients are depleted in the area where they're living, what happens is the amoeba swarm together. They start moving together, and you can see that where cell streams at around 6 hours. The single cells start moving together, they aggregate, they form this another kind of a structure, and they end up evolving or transforming, I should say, into a slug that migrates, a small worm. So these have gone from single individually existing cells to in a sense, a multicellular organism that now moves around. So people who are studying, let's say, the evolution of how single cells can turn into a multicellular entity are interested in looking at that process. Now, another thing happens, and that is they form multiple cell types. And what happens is, at some point, they form, the slug will differentiate into what's called a stalk and a fruiting body. And you can see pictures of this on the right-hand side. You see that long stalk, like a long pole-like structure with that ball at the top. So that's the stalk and the fruiting body. And what happens is it forms that, and then inside the fruiting body, spores form. And then when the fruiting body dries out, it cracks open, and the spores fly out, and they're carried by the wind to different places. And then they land on the ground, and they get wet, and then they differentiate back into amoeba. So here we have an example of a single cell going to a multicellular organism, and now it differentiates into different cell types. And so and there are very few cell types, so people who are using this organism are interested in how these different cells become different cell types, what genes are involved. So that's Dictyostelium, another model organism. So I want to tell you a little bit about Arabidopsis. I'm not a plant biologist, but anybody who is a plant biologist is actually using Arabidopsis as a model system. This is a relatively new model system that's been developed. Again, for the similar reasons as other model systems. They're easy to grow. They're small. You can grow them in small spaces. In fact, you can grow these Arabidopsis in small beakers or small cups with a little bit of soil. They go from seed to flower, I think, in 2 weeks. The whole genome has been sequenced, and they have all the organs of a regular plant. They have seeds, they have roots, they have flowers, they have stems, stalks, and so on. And so people are interested in how plants develop and how we can maybe modify plants to carry out different functions, use Arabidopsis as a model system. And we have one faculty member here in our department, Beth Krizak, who is interested in flower development and what genes are required for proper flower development. And she's made some really interesting findings regarding what genes interact in order to create a perfect flower organ. So that's an important plant model system. There are other model systems for plants. In the old days, people would use tobacco, they would use tomato, and some people still do, but this really is kind of the more well-developed system. The entire genome's been sequenced. So now I want to look at another multicellular organism. This is a fruit fly. Everybody's Drosophila melanogaster, and this is a fly that everybody has probably seen hanging around your bananas or your fruit that you have in the house for a little bit too long. They're tiny little flies. They have red eyes. I think they're kind of cute, in fact. This has been used as a model system for many different things for well over 100 years. There are multiple organs, so you can look at genes that are required for developing an eye, developing wing. People use it to look at effective environment on development, looking at behavior, looking at organ development. So it's used to study the genes that are involved in the development or the behavior or reproduction of a more complex organism. Now, some extremely interesting findings have been made using Drosophila. I'm going to tell you about a couple of those, particularly genes that are involved in the construction or the development of specific organs. Now here's one of those mutants. This is a Drosophila mutant called antennapedia. It's a mutation in what's called a homeobox gene. And there's many homeobox genes. Homeobox genes go from insects all the way to mammals, so we have them too. But the homeobox genes are master control genes that control the development of an entire organ, a single gene. So here's an example of antennapedia. And what happens with this particular mutant, if you look at the top of the head, normally at the top of the head there are antennae. What happens with the single gene mutation is that the antennae are converted to legs. So now these Drosophila have legs coming out of the top of their head. Why is this so important? What's so important is that it's a master switch that controls the whole developmental fate of an organ. And of course, the question is, what genes does this thing control that it would switch one organ to another? And of course, that's been pretty well studied. Here's another example. This is another example, maybe even a little bit more dramatic. This is called ultrabithorax. What happens with this particular mutation is that fruit flies usually only have two wings. Those are the two wings that are up closer to the top of the head. Below in the abdomen, what's normally there is called a haltere. There's one on each side. And halteres are kind of like gyroscopes, and they help keep the fly level during flight. They help with balance and help with leveling of the fly during flight. Now, a single mutation in this one homeobox gene called ultrabithorax changes the development of the haltere into an entirely new wing. So an entire organ is changed by one single mutation. And so again, it's a master regulator, and it gave us an enormous amount of insight in how organs are developed, and that a single gene can cause an entire conversion of one organ into another. Here's another model system that's used. This is a worm that exists in the soil. It's called C. elegans. The worm is very tiny. In fact, it only has 959 cells exactly, 959. It's transparent, so you can see right through it. It's about the size of an eyelash, so they're really tiny. You can't see them really well without a microscope. You can see every single cell, as I mentioned. These are useful because they have an entire reproductive system. They have an entire digestive system. People have been able to study cell division, how these different organs develop. They've turned out to be a very important model system for development in general. In fact, even people who are studying cancer biology, in some cases, have moved to look at C. elegans. Also looking at cell interactions, I'll talk about that in a minute or two. Now, in terms of the history of the development of this C. elegans as a model organism, I just want to tell you a little bit about an individual named Sidney Brenner. Sidney Brenner is a scientist who has been around for a long, long time. I think my guess is he's probably over 90 or close to 90 years old now. He's still active. Originally from Australia, he moved to England as a young man, went to college there, became connected with Watson and Crick and that whole group of people, and was extremely influential. In fact, he was one of the people who worked out the genetic code. And he also worked out the structure, worked out a lot about translation, a lot about tRNAs, some of the really early important work. And everybody thought at that time he was up for a Nobel Prize because deciphering the genetic code was pretty phenomenal. It ended up he didn't get a Nobel Prize, but he of course still remained very influential and famous in the field. And then a little bit later in life, he was working, I believe it was at Cambridge University, had his own lab, and he pretty much disappeared from the scene for about 10 years. And during those 10 years, he discovered the C. elegans, and he was working on developing it as a model system, meaning he was generating mutations in different genes and then looking at their effect and how it would affect, let's say, ovary development, or how it would affect the digestive system. So he developed a lot of mutants, and since that time, the field has grown enormously. But after 10 years of working alone, nobody really exactly knew what he was doing. He published one single paper. One paper, which became kind of the handbook of C. elegans biology. It described C. elegans as a model system. There are hundreds of labs around the world now using C. elegans as a model. And in fact, everybody traces their lineage, in a sense, back to Sidney Brenner. So I know people who studied with Sidney Brenner. I know people who studied with people who studied with Sidney Brenner, and so on. So kind of your status in the field is how close are you to this person. Well, after all that work and developing that model system, he ended up getting a Nobel Prize for the development of C. elegans as a model system. So even though he probably should have had 2 Nobel Prizes, but he didn't get it for the genetic code, but he got it. It became so important that he ended up getting the Nobel Prize for his development of this model system. Just want to show you one more slide on C. elegans. So this is another reason why it's so important, and that is, like I said, it's transparent. There's 959 cells. You can follow every single cell from fertilization until there's a complete organism. So this shows the lineage. So up at the very top, you can see the zygote. Zygote is fertilized, develops into 2 cells, which go to either end of those lines. And then differentiation and cell division continues, and you can see all of the lineage of every single cell type and what it ends up as. So what people have been able to do is, since it's clear, the body is clear, people have asked questions about what's the impact of cell-cell interaction. So 2 cells are together in development. Does that have any effect on what happens later, like what these cells differentiate into, or how efficiently they differentiate? And so what scientists have done is they've used lasers to kill single cells during development. So they target specific cells and ask, what's the result, or what happens to the neighboring cells? And in many cases, the differentiation of the organ becomes defective, telling us something about the requirement of proper cell-cell interaction. So it's been a very, very useful system. So let's go to another one. There's a few more, not too many. This is zebrafish. So this is the first time in my discussion of these model systems that we are looking at a vertebrate. So it turns out they're kind of similar to us. They have a well-developed vertebrae, they have a well-developed nervous system, well-developed eyes, well-developed reproductive system, digestive system. They actually have a olfactory system, that is, they smell. They detect smells in the water, and that's how they're attracted to the food. And in fact, the first odorant receptor, that is the receptor that can recognize a specific odor, was cloned and analyzed, was actually from zebrafish. But it's been used as a model for development of different organs. And there's lots of mutations, so you can look at the differentiation and the effect that those mutations have. We have 2 people in our department who are using C. elegans as a model system. One is Dr. Fabienne Poulain. She's a neurobiologist, and she uses the C. elegans. She uses the zebrafish as a model for how cells interact, how nerve cells interact during brain development. How does the brain form? And she has lots of different mutants that she grows. And in fact, it's interesting, if you go to her lab, she has a special room where they grow all these. And if you walk into that room, it's basically a dozen, 100 aquariums, small little aquariums, each containing a different mutant version of the zebrafish. There's another person, Dr. Katie Catherine, who also has a zebrafish colony, who uses that as the model system. And what she studies is blood development. So how does blood develop? What are the steps in blood development, and what genes are required for development of blood? So zebrafish, another important model. I'm going to show you another one now, which is really very bizarre, but it's kind of a new model system. It's not very pretty. So this is called an axolotl. It's like an amphibian, grows in the water, lives in the water. And the power of this particular organism, and the reason why people are studying it, is because it can regenerate every single organ in its body. If you cut off a leg, grows a new leg. If you cut off 2 legs, grows 2 legs. If you cut the body in half, it regrows the other half. So every single organ can be regenerated. And so people who are interested in studying the factors that are involved in regeneration and regeneration medicine are using axolotl as a model. We, of course, don't regenerate very much. The only organ that we can regenerate, and it's not clear why, is the liver. And you can cut out half your liver and it will eventually grow back. So we can regenerate liver, but that's it. And of course, people would like to learn about how that happens, what the process is, and maybe we can take advantage of that. So that's the odd-looking axolotl. And now we come to the lab mouse, Mus musculus. This is also another organism, model organism, that has been used, again, for well over 100 years. Here we're looking at a mammal. So we can study placental development, we can study fertilization, we can study behavior, all kinds of processes that are analogous to our processes can be studied in the mouse. In fact, we have about 75% genetic similarity with the mouse. We can introduce genes into the mouse. In fact, the lab mouse, Mus musculus, has been used to develop models for just about every single disease that there is. Just about every single disease, there is a mouse model that mimics it. So there's a mouse model for colon cancer, for all kinds of leukemias, for lymphomas, for breast cancer, for brain cancer, for cystic fibrosis, for Down syndrome. If you introduce the gene that is responsible for cystic fibrosis, the mice develop cystic fibrosis. If you introduce the gene that's responsible, or one of the genes that's responsible for colon cancer, they will develop colon cancer. And so that's important because it can be used, these mice are then used to study the disease, but more importantly, to study therapeutics and to develop drugs or treatments for these various syndromes. There's mouse models for Down syndrome. There's mouse models for anorexia. There's mouse models for autism. There's hundreds of them. And in fact, in order to develop any drug that's used for humans, you have to, by law, first test in mice. The FDA will not approve any drug or any treatment or any vaccine if it is not first tested in mice. In fact, the coronavirus vaccine that's being worked on now, there's actually I think 10 or 14 companies that are looking for vaccines. They're testing them in mice. They're testing them in monkeys as well. But there has to be some model system to test before you move into humans, and that is to test for the efficacy of the drug. In other words, does it work? To test for toxicity of the drug, to test for the dosage of the drug. All these things have to be worked out in great detail before moving into humans. So they're extremely important. Here's an example of a transgenic mouse. This is a transgenic mouse model for inherited obesity. And I think you can tell which one is the transgenic. The one on the right is a normal mouse. The one on the left has had a gene introduced for a particular growth factor that, when not functioning properly, causes this kind of obesity. And so these mice then can be used to test effective diet, effective different treatments, effective any drugs that may be developed. So again, the mouse is a very important model system. Finally, we want to come to people. So of course, a lot of what we do is we like to understand human biology, how we function, how diseases occur, treatments for diseases, treatments for any kind of behaviors that may be detrimental. But we cannot use humans as experimental organisms. It's not entirely true, because there are drug trials where where humans are used. Anytime there's a drug, let's say a new drug for let's say a cancer therapy, it first goes through the mouse studies to determine whether or not it would work or not, and the toxicity and so on. And if it passes all the approvals, then it goes into what are called clinical trials, where people sign up and volunteer to participate. So if we're talking about cancer, for example, there may be individuals who have tried every single thing there is to try, and basically there's nothing else to help them. And so they're willing to try a new drug. So those clinical trials are extremely important. Anything is used, any kind of drug needs to go through clinical trials to see how well it works. And then if it looks like it's successful, then it can go to the general public. So we do use humans in a sense, but of course it's with their permission. And speaking of the COVID and the coronavirus vaccine, so those vaccines are now moving from the mouse models and the monkey models, where it looks like, at least one of them looks like they're quite successful in generating some kind of antibody response, immune response. They are now moving into human trials, and they're basically asking for volunteers to test whether or not you generate a immune response, and then whether or not you would be resistant to infection. So we don't normally use human beings for our experiments, but if you want to work on human cancer, you want to work on human biology, human cells. So these represent human cells. These are cells that you can grow in culture. And most of the work that I do, we can't work with human beings. We're studying cancer. And the best we can do is work with these cells that you can grow in culture. There's lots of different cells that are growing in culture. People ask a lot of questions about cell division and how cells respond to different environmental insults, and let's say DNA damage and hormones. And so you can do those kinds of experiments. But you have to realize there are some caveats, there are some issues and some drawbacks using these cells. And I'll tell you what they are. The first is that most cells cannot grow in culture. There's a number of different cell types that cannot be grown in culture. We just don't know how. The other thing is that most normal cells will not grow for a long time in culture. They'll grow a little bit. I'll talk about this next time. So they grow for a little while and then they stop growing. So what people end up doing is they end up using cancer cells So most, if not all, the cells that people grow, scientists grow in the lab to do their work in cell biology are cancer-derived cells, because these cells grow indefinitely and you can do experiments forever. So that's one problem. They're not normal. Cancer cells are not normal, but in any case, it's kind of the best we can do. The second issue, the second caveat with using these human cells is they are not in any kind of physiologically normal context. They're taken out of the body, where they're in contact with other tissues, and put into culture in a plastic dish in an incubator with synthetic media. So that's not normal at all. So they're not in any kind of normal physiological condition, but yet they divide, they grow, they respond. They undergo cell division, and they carry out many of the processes that we want to study. And so even with those caveats and even with those questions, we still use them, and they still have been able to generate lots of important information. All right, so I'm going to stop here. We've looked at all these various model systems. Next time we're going to talk about human cells and how they grow in culture, a little bit about that. And so we will end it here. You have a nice rest of your day, and until next time.